Basics of Medical Biochemistry. First edition, 2022. Chapter 10. Metabolism of Carbohydrates, Part 2 of 3. By Professor Dr. Muhammad Jawad. MBBS, Tao, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 10.6 hexose monophosphate, HMP, shunt. It is an alternate pathway of glucose oxidation. It neither produces ADP nor utilizes it. It involves the utilization of all forms of monosaccharides with the liberation of CO2 and many side products such as NADPH plus H+. It is also known as the pentose phosphate pathway or phosphogluconate pathway. The pyruvate produced during glycolysis is shunted into the citric acid cycle and oxidative phosphorylation. However, there is an alternative cytosolic pathway that moves away from glycolysis and leads to the formation of the sugars necessary for nucleic acid, DNA and RNA, production. The HMP shunt utilizes a molecule that is produced in the first step of glycolysis, i.e., glucose 6-phosphate. It is used by the HMP shunt to generate NADPH, 5-carbon sugars known as pentoses, and ribose 5-phosphate, which serve as precursor molecules for nucleotide synthesis. NADPH plays an important functional role not only in HMP shunt but also in other biosynthetic processes, including fatty acid metabolism and control of reactive oxygen species, ROS. Salient features of the HMP shunt are: it takes place in the cytoplasm of different body organs. It is active in tissues that have metabolic and synthetic pathways such as the liver, adipose tissue, lactating mammary glands, RBCs, suprarenal cortex, thyroid, ovary, and testis. Its activity is low in the non-lactating mammary gland, and skeletal muscles. It functions for producing NADPH plus H plus and ribose. It does not mean to produce ADP. The H plus acceptor in this pathway is NADP. 10.6.1 Phases of HMP shunt. There are two distinct phases in this pathway: I oxidative phase in which NADPH plus H plus is generated. 2 non-oxidative phase in which 5-carbon sugars are synthesized. 10.6.2 Reactions of HMP shunt. In the HMP shunt, 3 molecules of glucose 6-phosphate give rise to 3 molecules of CO2 and 3 molecules of 5-carbon sugars, these rearranged to produce 2 molecules of glucose 6-phosphate. Following are the reactions of the HMP shunt. 1. Dehydrogenation of glucose 6-phosphate to 6-phosphogluconolactone. The first reaction is the oxidation of glucose 6-phosphate. Glucose 6-phosphate is converted into 6-phosphogluconolactone by the enzyme glucose 6-phosphate dehydrogenase. It is the rate-limiting step. 2. Conversion of 6-phosphogluconolactone to 6-phosphogluconate. In this reaction, hydration of 6-phosphogluconolactone by the enzyme lactonase converts it into 6-phosphogluconate. 3. Formation of ribulose 5-phosphate. Dehydrogenation of 6-phosphogluconate converts it into ribulose 5-phosphate. The enzyme is 6-phosphogluconate dehydrogenase and the coenzyme is NADP+. 4. Formation of ribose 5-phosphate. It is formed by the isomerization of ribulose 5-phosphate. The enzyme is phosphopentose isomerase. 5. Formation of xylulose 5-phosphate. It is formed by epimerization of ribulose 5-phosphate. The enzyme is epimerase. 6. Formation of glyceraldehyde 3-phosphate and cytoheptulose 7-phosphate. These are formed by the reaction between ribose 5-phosphate and xylulose 5-phosphate. This is called transcytolation, which takes place by the enzyme transcytolase. 7. Formation of fructose 6-phosphate and erythrose 4-phosphate. These are formed by the interaction between cytoheptulose 7-phosphate and glyceraldehyde 3-phosphate by the enzyme transaldolase. 8. Formation of fructose 6-phosphate and glyceraldehyde 3-phosphate. These are formed by the interaction between xylulose 5-phosphate and erythrose 4-phosphate by the enzyme transketolase. 10.6.3 Regulation of HMP shunt. The glucose 6-phosphate dehydrogenase is the key regulatory enzyme. It is activated by fed state, glucose, insulin, and NADP. But it is inhibited during starvation, diabetes mellitus, and with a high NADP plus H plus /NADP ratio. 10.6.4 Functions of HMP shunt. There are two main functions of the HMP shunt. 1. Production of pentoses for the synthesis of nucleosides, nucleotides, nucleic acids, ADP, coenzymes, etc. 2. Production of NADP plus H plus that is required for regeneration of reduced glutathione, lipogenesis, cholesterol, and steroid synthesis. Synthesis of tyrosine from phenylalanine. Metabolic hydroxylation with CYP450. As coenzyme for methemoglobin reductase. Synthesis of sphingosine and cerebrosides. Synthesis of fructose from glucose. Figure 10.11 Structure of glutathione, a tripeptide composed of gamma-glutamate, cysteine and glycine. Figure 10.12 Reactions of hexose monophosphate, HMP, shunt. 10.6.5 Mechanism regarding the role of NADP plus H plus in the protection of RBCs. In erythrocytes, NADP plus H plus produced from the HMP pathway is used to regenerate GSH, glutathione in reduced form, from its oxidized form by glutathione reductase. In turn, reduced glutathione removes hydrogen peroxide, H2O2, from the erythrocyte in a reaction catalyzed by glutathione peroxidase. H2O2 is a toxic substance produced due to metabolism or toxins, which can decrease the lifespan of the erythrocyte by causing oxidative damage to the cell membrane leading to hemolysis. Glutathione reductase. GSSG plus NADP plus H plus NADP plus 2-GSH. Oxidized glutathione, reduced glutathione. Glutathione peroxidase. H2O2 plus 2-GSH GSSG plus 2H2O. Glutathione peroxidase catalyzes the degradation of organic hydroperoxides by reduction, as two glutathione molecules, represented as GSH, are oxidized to a disulfide. Glutathione peroxidase uses the trace element selenium as a functional group. The enzyme's primary structure includes an analog of cysteine, selenocysteine, with SC replacing S. 10.7 Glycogen metabolism. Glycogen is a highly branched polymer of alpha-D-glucose having both alpha, 1 rightward arrow 4, glycosidic bond in a linear chain and alpha, 1 rightward arrow 6, glycosidic linkage at the branch point. Glucose is stored as glycogen predominantly in liver and muscle cells. Glycogen is abundant in the liver, 100 grams, comprising 8 to 10% of the fresh mass of well-fed liver in adults. About 400 grams of glycogen is present in skeletal muscles which make up 1 to 2% of their mass. Liver glycogen is responsible to maintain blood glucose levels, particularly during the early stages of fasting. Muscle glycogen is regarded as a fuel reserve for the synthesis of ADP during muscle contraction. Why is glycogen stored and not glucose? Glycogen is not soluble in water and cannot pass in and out of cells unless it is broken down into smaller, more soluble units of glucose. Its usefulness as a storage molecule is largely due to this insolubility. Moreover, glycogen exerts little osmotic pressure and does not disturb the intracellular fluid balance. Metabolism of glycogen comprises glycogenesis and glycogenolysis. 10.7.1 Glycogenesis. It is the process of glycogen synthesis. Glycogenesis is an anabolic process that requires ADP energy to assemble excess glucose molecules into more complex glycogen granules. A single glycogen granule can contain 30,000 glucose units. Glycogenesis takes place when blood glucose levels are sufficiently high to allow excess glucose to be stored in liver and muscle cells. The main steps of glycogenesis are 1. Phosphorylation of glucose, 2. Formation of glucose 1-phosphate, 3. Formation of uridine diphosphate glucose, UDP glucose, 4. Formation of amylose chain and 5. Formation of the branch point. 1. Phosphorylation of glucose. This step is the same as the first step of glycolysis, during which glucose is phosphorylated to glucose 6-phosphate. 2. Formation of glucose 1-phosphate. Glucose 6-phosphate is converted to glucose 1-phosphate in a reversible reaction catalyzed by phosphoglucomutase. Glucose 6-phosphate glucose 1-phosphate. 3. Formation of uridine diphosphate glucose, UDP glucose. It is formed by the reaction between glucose 1-phosphate and uridine triphosphate, UTP, by the enzyme UDP glucose pyrophosphorylase. Glucose 1-phosphate plus UTP UDP glucose plus PPI. Uridine diphosphate glucose, UDP glucose, is the immediate precursor for glycogen synthesis. As glucose residues are added to glycogen, UDP glucose is the substrate and UDP is released as a reaction product. Nucleotide diphosphate sugars are also the precursors for the synthesis of other complex carbohydrates, including oligosaccharide chains of glycoproteins, etc. Figure 10.13 Formation of uridine diphosphate, UDP, glucose, UTP, uridine triphosphate, PPI, pyrophosphate. 4. Formation of amylose chain. The UDP glucose complex has an affinity for primer molecules. Primer is an already existing fragment of glucose and it must be required for the synthesis of glycogen. UDP glucose complex is bound with primer in such a way that glucose is transferred to the hydroxyl group at AC4 terminal, non-reducing end, of primer to form alpha, 1 rightward arrow 4, glycosidic linkage. In this elongation reaction, UDP is displayed by the terminal hydroxyl group of the growing glycogen molecule. This reaction is catalyzed by glycogen synthase. Glycogen, N residues, plus UDP glucose glycogen, N+1 residues, plus UDP. UDP released from the UDP glucose complex is then phosphorylated by ADP in a reaction catalyzed by nucleoside diphosphate kinase. UDP plus ADP UTP plus ADP. Newly synthesized UTP is reacted with another molecule of glucose 1-phosphate and formed UDP glucose to grow glycogen, but in this type of synthesis, glucose molecules are linked through alpha, 1 rightward arrow 4, glycosidic linkage to form an amylose chain. The process is continued till the point of branching. Role of glycogenin in glycogen synthesis. Glycogen synthase can work only in the presence of glycogen primer. The first-time formation of glycogen is carried out with the help of glycogenin. Glycogenin is an enzyme that catalyzes glycosylation of one of its own tyrosine residues, that is amino acid number 194. A glycosidic bond is formed between the anomeric C1 of the glucose moiety derived from UDP glucose and the hydroxyl oxygen of a tyrosine side chain of glycogenin. Glycosylation at C4 of the O-linked glucose product yields an O-linked disaccharide with alpha, 1 rightward arrow 4, glycosidic linkage. UDP glucose is again the glucose donor. This is repeated until a short linear glucose polymer with alpha, 1 rightward arrow 4, glycosidic linkages is built up on glycogenin. UDP is released as a byproduct. Figure 10.14 Role of glycogenin in glycogen synthesis. 5. Formation of branch point. Branching occurs when the amylose chain grows to at least about 11 glucose residues by glycogen synthetase. For the formation of a branch, another enzyme is required to form alpha, 1 rightward arrow 6, glycosidic linkage, called branching enzyme, also known as amylo-alpha, 1 rightward arrow 4, rightward arrow alpha, 1 rightward arrow 6, transglycosylase. The branching enzyme transferred a set of 6 to 8 glucosyl residues from the non-reducing end of the glycogen chain by breaking alpha, 1 rightward arrow 4, glycosidic bond and attached it to a more interior site by making alpha, 1 rightward arrow 6, glycosidic linkage. As a result, a new non-reducing end is created on the branch along with the original one from where the fragment of 6 to 8 residues was removed. These both non-reducing ends are further elongated by glycogen synthase. Branching gives glycogen two advantages over starch as a storage form of glucose. First, it is more soluble than its unbranched structure. Second, the exposure of more C4, non-reducing, ends means that glycogen can be both synthesized and degraded more quickly than a single starch chain with the same number of residues. Figure 10.15 Steps of Glycogenesis, UDP, uridine diphosphate. 10.7.2 Glycogenolysis. It is the catabolism of glycogen during which the breakdown of glycogen to glucose units takes place. There are two main steps of glycogenolysis, 1. Shortening of the chain and 2. Removal of the branch point. 1. Shortening of chain. Glycogen phosphorylase catalyzes phosphorylitic cleavage of the alpha, 1 rightward arrow 4, glycosidic linkages of glycogen, releasing glucose 1-phosphate as the reaction product. Glycogen, N residues, plus pi glycogen, N, 1 residues, plus glucose 1-phosphate. Figure 10.16 Action of glycogen phosphorylase and debranching enzyme during glycogenolysis. Glycogen phosphorylase can sequentially cleave alpha, 1 rightward arrow 4, linkages on non-reducing ends of the glycogen chain until 4 residues remain before the branch point, i.e., alpha, 1 rightward arrow 6. After the shortening of all branches of the glycogen molecule, the structure left is called a limit dextrin. Limit dextrin is then attacked by the debranching enzyme. The coenzyme for glycogen phosphorylase is pyridoxal phosphate, PLP. 2. Removal of branch point. Debranching enzyme is responsible for the removal of branch point during glycogenolysis. It has 2 independent active sites, consisting of residues in different segments of a single polypeptide chain. The transferase i.e., oligo-alpha, 1 rightward arrow 4, rightward arrow alpha, 1 rightward arrow 4, glucan transferase of the debranching enzyme transfers 3 glucose residues from a 4 residue limit branch to the end of another branch, diminishing the limit branch to a single glucose residue. 2. The amylo-alpha, 1 rightward arrow 6, glucosidase moiety of the debranching enzyme. It catalyzes the hydrolysis of the alpha, 1 rightward arrow 6, linkage, yielding free glucose. This is a minor fraction of glucose released from glycogen. The remaining molecule of glycogen is again available for the action of glycogen phosphorylase and debranching enzyme to repeat the reactions. Through the combined action of glycogen phosphorylase and debranching enzyme, glucose 1-phosphate and free glucose are produced in a ratio of 8:1. Glycogenolysis in lysosomes. Small amount of glycogen, 1 to 3%, is continuously degraded by a lysosomal enzyme called alpha, 1 rightward arrow 4, glucosidase, acid maltase. Deficiency of this enzyme results in glycogen accumulation, causing glycogen storage disease type 2, Pompe's disease. Figure 10.17 Steps of glycogenolysis. The fate of glucose 1-phosphate. It is converted to glucose 6-phosphate by the action of the enzyme phosphoglucomutase. It catalyzes the following reversible reaction. Glucose 1-phosphate, glucose 6-phosphate. The O group of serine at the active site of phosphoglucomutase donates and accepts pi. The fate of glucose 6-phosphate in the liver, it is either changed into I-glucose by glucose 6-phosphatase or 2-fructose 6-phosphate by the enzyme isomerase goes into glycolysis. Liver glucose 6-phosphatase catalyzes the following reaction, essential to the liver's role in maintaining blood glucose. Glucose 6-phosphate plus H2O glucose plus pi. This reaction takes place in the endoplasmic reticulum of hepatocytes. Glucose 6-phosphate is transported to the endoplasmic reticulum by glucose 6-phosphate translocase. Glucose 6-phosphatase is absent in muscle, brain, and most other tissues, hence free glucose cannot be produced from glucose 6-phosphate in these tissues. The fate of glucose 6-phosphate in muscles. Glucose 6-phosphate in muscles usually follows glycolysis, however, it can give glucose indirectly through the Cori cycle. 10.7.3 Regulation of glycogen metabolism. Glycogenesis and glycogenolysis are controlled by the enzymes glycogen synthase and glycogen phosphorylase. There are 3 mechanisms to regulate glycogen metabolism: allosteric regulation, hormonal regulation, and the influence of calcium. 1. Allosteric regulation. There are certain metabolites that allosterically regulate the activities of glycogen synthase and glycogen phosphorylase. The glycogen synthesis is increased when substrate availability and energy levels are high. Glycogen breakdown is enhanced when glucose concentration and energy levels are low. In a well-fed state, the availability of glucose 6-phosphate is high, which allosterically activates glycogen synthase for more glycogen synthesis. Glucose 6-phosphate and ADP allosterically inhibit glycogen phosphorylase. Free glucose in the liver also acts as an allosteric inhibitor of glycogen phosphorylase. 2. Hormonal regulation. Hormones, through a complex series of reactions, bring about covalent modification, namely phosphorylation and dephosphorylation of enzyme proteins which, ultimately control glycogen synthesis or its degradation. The hormones like epinephrine, norepinephrine, and glucagon, in the liver, activate adenylate cyclase to increase the production of CAMP. The enzyme phosphodiesterase breaks down CAMP. The hormone insulin increases the phosphodiesterase activity in the liver and lowers the CAMP levels. Effects of hormones on glycogenesis. Glycogen synthase is the key enzyme in glycogenesis. Glycogenesis is primarily regulated by modulating the activity of glycogen synthase. This enzyme exists in two forms: dephosphorylated, active or A form, and phosphorylated, inactive or B form. It is regulated by covalent modification, in an inverse direction to that of glycogen phosphorylase. The phosphorylation of glycogen synthase is catalyzed by various protein kinases in response to CAMP. The dephosphorylation of glycogen synthase is catalyzed by the protein phosphatase 1, pp1. Glycogenesis is stimulated by the hormone insulin in response to hyperglycemia and inhibited by epinephrine. The inhibition of glycogen synthesis is brought by epinephrine, also norepinephrine, and glucagon through CAMP by converting active glycogen synthase A to inactive form, i.e., glycogen synthase B. Effects of hormones on glycogenolysis. The hormones like epinephrine and glucagon bring about glycogenolysis by their action on glycogen phosphorylase through CAMP. Glycogen phosphorylase exists in two forms, an active A form which is phosphorylated and another inactive form B which is dephosphorylated state. The CAMP activates CAMP-dependent protein kinase. Protein kinase phosphorylates the inactive form of glycogen phosphorylase kinase to the active form. The enzyme protein phosphatase removes phosphate and inactivates phosphorylase kinase. An elevated glucagon or epinephrine level increases glycogen degradation, whereas an elevated insulin results in increased glycogen synthesis. 3. Influence of calcium. When the muscle contracts, Ca2+ ions are released from the sarcoplasmic reticulum. Released Ca2+ binds to calmodulin, a calcium-modulating protein, and directly activates phosphorylase kinase without the involvement of CAMP-dependent protein kinase. Figure 10.18 Regulation of glycogen metabolism through hormone-mediated covalent modification of enzymes, pKa, protein kinase A. Source: Lipinkat's Illustrated Review Biochemistry 7th edition. 10.8 Glycogen storage diseases or glycogenoses. These disorders are due to the congenital absence or deficiency of certain enzymes involved in carbohydrate metabolism. These are associated with excessive accumulation of normal or abnormal glycogen within cells. A brief description of some important glycogen storage diseases is given in Table 10.6. Table 10.6 Glycogen storage diseases. Type of glycogen storage. Disease and enzyme deficient. Symptoms. In addition to the accumulation of glycogen. Type 0, deficiency of glycogen. Synthase. Accumulation of abnormal glycogen. Hypoglycemia. Hyperketonemia and early death. Type Ia, von Gierke's disease. Deficiency of liver and kidney glucose. 6-phosphatase. This disease is named after Edgar von Gierke, the German doctor who discovered it. An increased amount of glycogen with normal structure. Doll-like face with excess fat in the cheeks. Massive. Enlargement of the liver, severe fasting hypoglycemia. Ketosis, hyperuricemia, and hyperlipidemia. Growth failure. and progressive renal disease. Treated by nocturnal gastric infusion of glucose or regular use of uncooked starch. Type IB, von Gierk's disease. Deficiency of glucose. 6-phosphate translocase. Same as type IA plus neutropenia and recurrent infections. Type 2, Pompe's disease. Deficiency of lysosomal alpha, 1 rightward arrow 4, glucosidase, acid maltase, in the liver, kidney, heart, and muscles. Excessive glycogen of normal structure in lysosomes. Bulky muscles include macroglossia, enlarged tongue, and cardiomegaly, hypotonia, and muscle weakness, including congestive heart failure, cardiorespiratory failure. Causes death, usually before 2 years of age. Treated by enzyme replacement therapy. Type 3A, 4B's or Cory's disease or limit dextrinosis deficiency of debranching enzymes in muscles and liver. Fasting hypoglycemia, hepatomegaly in infancy. Accumulated glycogen with short outer branches. Limit dextrin, and muscle weakness. Type IIIB, limit dextrinosis. Deficiency of debranching enzyme in the liver. As type 3A but without muscle weakness. Type 4, Anderson's disease. Or amylopectinosis deficiency of amylo alpha, 1 rightward arrow 4, rightward arrow alpha, 1 rightward arrow 6. Transglycosylase, branching enzyme. The affected organs are the liver, muscles, heart, brain and spleen. Progressive cirrhosis, liver or heart failure causes death, usually before age 5. Type V, McArdle's disease. Deficiency of muscle glycogen phosphorylase. This disease was first reported in 1951 by Drive. Brian McArdle of Guy's Hospital, London. An excessive amount of glycogen in muscle with normal structure. Unable to perform strenuous exercise because of painful muscle cramps otherwise normal. Myoglobinemia and myoglobinuria is also present. Type 6, HER's disease. Deficiency of liver glycogen phosphorylase. Like type I but with a milder course. Type 7, Taruy's disease. Deficiency of phosphofructokinase 1 in muscle and RBCs. Poor exercise tolerance is associated with low lactate after exercise. Hemolytic anemia also found. Type 8, deficiency of phosphorylase kinase in the liver. Required to activate glycogen phosphorylase. Mild liver enlargement, mild hypoglycemia. Symptoms abate after puberty with proper treatment. 10.9 Metabolism of fructose. Fructose is present in fruit juices and honey. The chief dietary source of fructose is sucrose. Sucrose is hydrolyzed in the intestine by the enzyme sucrase. Fructose is absorbed by facilitated transport and taken by portal blood to the liver. It is mostly converted to glucose. The first event in the metabolism of fructose is its phosphorylation. Fructose is phosphorylated either by hexokinase or fructokinase. 1. Phosphorylation of fructose by hexokinase. Fructose is phosphorylated to fructose 6-phosphate by the enzyme hexokinase. The affinity of the hexokinase for fructose is very low as it has high kilometer, therefore, little fructose is phosphorylated by this enzyme. This reaction takes place mainly in the kidney and muscles. Fructose 6-phosphate may enter the glycolytic pathway or be converted to glucose or glycogen. Figure 10.19 Outline of fructose metabolism. 2. Phosphorylation of fructose by fructokinase. Fructose is mostly phosphorylated by fructokinase to fructose 1-phosphate. Fructokinase is present in the liver, kidney, muscle, and intestine. It has a high Vmax and low Km for fructokinase. 3. Cleavage of fructose 1-phosphate. It is carried out by the enzyme aldolase B, fructose 1-phosphate aldolase. As a result, fructose 1-phosphate is broken down into two trioses, dihydroxyacetone phosphate and glyceraldehyde. Dihydroxyacetone phosphate is used either in glycolysis or gluconeogenesis. Glyceraldehyde is phosphorylated to glyceraldehyde 3-phosphate or reduced to alcohol. These products are then utilized in many metabolic pathways. Figure 10.20 Metabolism of fructose, VLDL, very low density lipoprotein. Metabolism of fructose is somewhat better than glucose in diabetes mellitus as it is not affected by insulin or its deficiency. Similarly, the rate of fructose metabolism is far more rapid than glucose as the effect of phosphofructokinase 1 is bypassed, which is the main rate-limiting step of glycolysis. Clinical aspects/defects of fructose metabolism. The metabolic disorders of fructose result from a deficiency of one of the key enzymes involved in its metabolism. Essential fructosuria, it occurs due to a deficiency of the enzyme hepatic fructokinase. In this condition, fructose is not converted to fructose 1-phosphate and there is an excretion of fructose in urine. Urine gives positive Benedict's and Selowanoff's tests. Treatment is the restriction of dietary fructose. Hereditary fructose intolerance, it is caused by a deficiency of aldolase B. It is an autosomal recessive inborn error. In this condition, fructose 1-phosphate is not metabolized. This disorder is characterized by intracellular accumulation of fructose 1-phosphate, which inhibit glycogen phosphorylase. There occurs accumulation of glycogen in the liver with associated hypoglycemia, vomiting, hyperuricemia and lactic acidemia. The main clinical features are liver damage and jaundice that can progress to cirrhosis and ascites. There is decreased gluconeogenesis as well as low protein synthesis and decreased absorption of inorganic phosphorus due to renal tubular damage. Management includes the removal of sucrose and fructose from the diet to minimize the chances of hepatic failure and expected death. 10.10 Metabolism of galactose. Galactose is an aldohexose. It does not occur free in nature. Inside the body, it is found as a constituent of milk, in the form of lactose in combination with glucose. It is also synthesized in the body where it forms a part of glycolipids and glycoproteins in several tissues. Lactase, beta-galactosidase, of intestinal mucosal cells hydrolyses the lactose to galactose and glucose. Galactose is also produced from the lysosomal degradation of glycoproteins and glycolipids. Galactose is metabolized almost exclusively by the liver and therefore galactose tolerance test is done to assess the functional capacity of the liver. UDP galactose is the active donor of galactose during synthetic reactions. Galactose passed through the following steps during its metabolism. 1. Galactose is first phosphorylated by galactokinase to galactose 1-phosphate. 1-ADP is utilized in this reaction. Galactose plus ADP galactose 1-phosphate plus ADP. 2. Galactose 1-phosphate reacts with UDP, uridine diphosphate, glucose to form UDP galactose and glucose 1-phosphate by the enzyme galactose 1-phosphate uridyl transferase. This is the rate-limiting enzyme. The glucose 1-phosphate is either dephosphorylated or isomerized to glucose 6-phosphate and enters glycolysis. Galactose 1-phosphate plus UDP glucose UDP galactose plus glucose 1-phosphate. Galactose 1-phosphate can also react with UTP to form UDP galactose by the enzyme UDP galactose pyrophosphorylase. This hepatic enzyme is at low levels during infancy. It gradually increases as age advances. Therefore, a galactosemic adult can handle the metabolism of fructose efficiently if he has been managed properly during the early age of infancy. 3. UDP galactose is isomerized to its C4 epimer i.e., UDP glucose by UDP hexose 4 epimerase. The newly formed UDP glucose may participate in glycogenesis or step 2 of galactose metabolism. Galactose is not an essential nutrient since UDP glucose can be converted to UDP galactose by the enzyme UDP hexose 4 epimerase. UDP galactose UDP glucose. 4. UDP galactose is an active donor of galactose. UDP galactose is essential for the formation of compounds like lactose, glycosaminoglycans, glycoproteins, cerebrosides, and glycolipids. Figure 10.21 Metabolism of galactose, UDP, uridine diphosphate, UTP, uridine triphosphate, P, phosphate, PPI, pyrophosphate. Lactose synthesis. Synthesis of lactose takes place by the reaction between UDP galactose and glucose. It is catalyzed by the enzyme lactose synthase or UDP galactose glucose galactosyl transferase richly present in lactating breast. UDP galactose plus glucose lactose plus UDP. Conversion of galactose to galactitol. Galactitol, dulcitol, is a sugar alcohol, the reduction product of galactose. It is produced from galactose in a reaction catalyzed by aldose reductase. This reaction occurs in the kidney, retina, lens of the eye, nervous tissue, seminal vesicles, and ovaries. Galactose plus NADP plus H plus galactitol plus NADP plus. Under a normal physiological state, galactitol is unimportant. But in people with galactosemia, excess galactitol forms in the lens of the eye leading to cataracts. Clinical aspects. Galactosemia, it is an inherited disorder representing the raised level of galactose in the blood. It is an inborn error of metabolism due to a deficiency of the enzyme required for the conversion of galactose into glucose. In classical galactosemia, galactose 1-phosphate and uridyl transferase are deficient, causing accumulation of galactose and galactose 1-phosphate in the body. Galactose is shunted to produce the respective alcohol known as galactitol or dulcitol. In this condition, galactose also appears in the urine. Symptoms. Newborn with galactosemia first appears normal at birth, but when they are given milk for the first time, symptoms appear. These symptoms appear on the third day of life. These are vomiting and jaundice. After 2 to 3 months, symptoms associated with galactosemia become more severe and include liver disorders, cataract formation, mental retardation, severe diarrhea. Diagnosis. Galactosemia can be diagnosed in utero by amniocentesis. In positive cases, exclusion of lactose and galactose from her diet is advisable. Important diagnostic features include increased blood galactose levels, increased sugar levels, increased excretion of galactose in the urine, albuminuria and aminoaciduria. Treatment. Lactose and galactose-free diet is the treatment of galactosemia. 10.11 Sorbitol metabolism. Sorbitol is produced in the body from glucose by the enzyme aldose reductase. Glucose plus NADP plus H plus sorbitol plus NADP plus. Sorbitol is converted to fructose in certain tissues of the body by the enzyme sorbitol dehydrogenase. Sorbitol plus NAD plus fructose plus NADH plus H plus. The above reaction occurs in the liver and seminal vesicles. In the liver, sorbitol is used for energy, but in seminal vesicles, it provides fructose to sperms, as it is the preferred source of energy for them. The system of aldose reductase and sorbitol dehydrogenase provides a way for the body to produce fructose from glucose without using ADP. Clinical aspects. Sorbitol pathway is rapid in uncontrolled diabetes. In uncontrolled diabetes, large amounts of glucose enter the cells which are not dependent on insulin. The cells with increased intracellular glucose levels in diabetes, lens, retina, nerve cells, kidney, etc., possess high activity of aldose reductase and sufficient supply of NADP. This results in the rapid and efficient conversion of glucose to sorbitol. The enzyme sorbitol dehydrogenase is either low in activity or absent in these cells. Hence, sorbitol is not converted to fructose. Sorbitol cannot freely pass through the cell membrane and accumulate in the cells. Sorbitol, due to its hydrophilic nature, causes osmotic effects leading to swelling of the cells. Pathological changes associated with uncontrolled diabetes are due to the accumulation of sorbitol. It is to be noted that continuous use of NADP during sorbitol production causes a decrease in the generation of reduced glutathione, which further aggravates the complications of diabetes. Sorbitol can be used as a laxative when taken orally or as an enema. Sorbitol works as a laxative by drawing water into the large intestine and stimulating bowel movements. Exercise questions. For MCQ 1 to 5 see Part 1 of this chapter and for 11 to 16 see Part 3 of this chapter. MCQ 6. Which of the following enzymes is responsible to trap glucose within the cell by catalyzing the phosphorylation of glucose to glucose 6-phosphate? Hexokinase. Glyceraldehyde 3-phosphate dehydrogenase. Phosphofructokinase. Pyruvate kinase. Phosphotriose isomerase. MCQ 7. Formation of which of the following substances is affected in the TCA cycle if the reaction between acetyl-CoA and oxalostate is inhibited? Citrate. Fumarate. Malate. Oxalostate. Succinate. MCQ 8. A 38-year-old man suffering from angina pectoris was brought to the hospital with complaints of chest tightness and pain on exertion. The pain subsides on rest. Which of the following metabolic processes is the main source of energy for the affected organ during ischemia in this patient? Cori cycle. Fatty acid oxidation. Glycogenolysis. Glycolysis. HMP shunt. MCQ 9. Which of the following biochemical pathways occurs partly in the mitochondria and partly in the cytosol? Gluconeogenesis. Glycogenesis. Glycogenolysis. Glycolysis. Ketogenesis. MCQ 10. Which of the following metabolic product is produced under normal circumstances by erythrocytes and muscle cells during intense exercise, that is recycled in the liver via the Cori cycle? Citrate. Glycerol. Lactate. Oxalostate. Pyruvate. Please write answers in the comments or contact drmjod@yahoo.com. End of Part 2 of 3 of Chapter 10. Thank you.